All right, what was going on? What was that all about? That's Yuki Wang, ladies and gentlemen. Please give him another hand. I will explain what that's about within a minute, so just be patient with me. I think the sense of touch is one of the most amazing senses. It is very diverse, it's very unique, and it's very profound. Compared to other senses, vision, smell, taste, hearing, which are easily defined, the sense of touch is a bit more different. What I mean by that is, so for example, if you think of the fifth sense, of course, touch is normal, but it also includes, if you define it more generally, sense of temperature, sense of pain, sense of your internal organs, we call it interoception. In scientific terms, we sometimes call this somatosensation. And what keeps them all together is that they sense physical stimuli, pressure or temperature, and translate it into chemical signals that the neurons can understand. But my favorite sense within this whole idea of somatosensation is actually proprioception. So what is this? I call it the most important sense that you have that probably many of you didn't even know that you had it. What it really is that as you move around, your muscles get stretched. And from how much your muscles are stretched, you have a very clear idea of where your limbs are in space. I can, for example, close my eyes and know exactly where my fingertips are and touch my nose. I can walk, coordination without looking. You can do tightrope walking like this image shows, or perhaps most impressively, play an amazing piece of music like Yuki just did, blindfolded. So why do we not know about this, and why do we take it for granted? I think mainly because you can't turn it off. Like, you can close your eyes and imagine a world without sight, but you can't turn proprioception off. And so it's always there. So we're going to talk about this. But coming back to the tactile skin touch sensation, it's really amazing, the sensitivity. So you can sense indentations in your skin that is 1/500th diameter, 1/500 diameters of the human hair. So take the human hair, divide it into 500 times, that little of indentation in your skin, and you can feel it. But literally, it's not just about sensitivity, there's quality too. And of course, touch is very pleasant, and we call this affective touch. And we know that without it, your emotional health is really relying on this sense of touch. But you know, I think this picture, to me, shows the power of touch more than any other. So this little guy is visiting the White House, and he's very shy, and he tells the president that, I heard that other kids tell me that my hair looks like yours. And President Obama just bends down and says, touch it, dude. And so we often say seeing is believing, but clearly touching is believing as well. And I'm not just talking about the texture of his hair. Once again, that affective touch, the pleasantness of touch, for this boy to feel like someone that looks like him, that his hair feels like his, is the President of the United States, is a very powerful message. So the flip side of touch is, of course, pain. And this is a beautiful painting by Frida Kahlo. She suffered through chronic back pain, and this is her imagination or telling you how pain feels to her. So pain, acute pain itself, is absolutely essential for life. But chronic pain, we usually define chronic pain as pain that lasts after the stimulus that caused it is gone away. And that's a major unmet medical need. People suffering from neuropathic pain, we don't have great medications for it. And so it's a very important medical field as well. And pain is complicated. It's a sensation as well as an emotion. So all of these, touch, proprioception, temperature sensation, pain, how do you sense this? You have neurons on two sides of your backbone next to the spinal cord. And these little balls that you see, I think this works. I went backwards instead of using the lighter. These balls that you see are groups of about 8,000 neurons that send these tiny little processes. We call them neurites. They look like little antenna, and they innervate every part of your body. They get information from there, take it to the spinal cord and the brain, and it's through these neurons. This is a zebrafish embryo, a video of it developing. This network of sensors tell you all this information that we talked about. So if we now think more from a molecular point of view, genes and proteins, how do you do this? How do you sense protein? The thing that was in common to everything I said, sensing touch, proprioception, pain, is all about sensing pressure. So we knew for a long, long time that there are these specialized molecules called ion channels that sit outside the cell. The cell is surrounded by lipid bilayer, this fatty thing that protects the cell. But to communicate with the outside, there's this protein that sit there and do different things. The ones that we care about is there should be pressure sensing, in the sense that if that membrane is stretched, you want them to open and let ions in. And this is the language of neurons. If there's ions coming in, it's like little electricity going into the neuron, and that's enough to start the whole signal to tell the brain what's going on. But what is the identity of this protein was unknown for the longest time, and that's what my lab was studying. So how do we find this? Initially, we tried to find it directly from neurons, but that proved to be very difficult. So in science, sometimes we say we take a reductionist point of view. So we found a cell that we can grow in a dish, in a culture, in the lab, that was mechanosensitive, and we found the protein within this. So what you're looking at is actually the assay that we use to measure this. There are two glasses involved. What you're seeing here is the cell. With this glass probe, we're actually poking the cell, giving it some little pressure. And on this glass, we're actually recording the electric current that this pushing would cause if it had this ion channel. Now, I'm not going to go through how we did and what we found, but we did find the gene or the protein that is responsible for pressure sensing. And this is what it looks like. This is the structure of the protein that we have resolved, and it's a thing of beauty. So it has three identical members here drawn in three different colors that come together. So in the center is that when, schematically I showed is the pore where the ions would go through. But the characteristic feature of this protein is all these little ribbons, we call them helices, that actually are used to extend into the plasma membrane. And this is kind of how they sense it. And you can see it has this bizarre puckered kind of dimension, like the cell's supposed to look like this, and this protein is looking like this. Very unusual for a protein to be that way. But we figured out that that's actually how it's pressure sensitive. And I'll show you in the next slide. So what we in the field, us and others, have found out that when you stretch the cell, this shape that's closed up actually opens. It stretches. It's like a little molecular machine that can change shape, and when it opens, you get these ion channels coming through. Okay, so now I have slides. I can easily show you this mechanism, but a few months ago I thought, what if I was in a bar talking to someone, and I love talking about my science, and I wanted to show how this works, what would I do? So this is like a little bit of performance art in the middle of science. I'm going to ask Yuki to hold my jacket for a second. And so I thought the best way to do this was to get a tattoo of this protein so that I can demonstrate it without slides. And so here it is. Thank you. And it's instructional, although my institute has not reimbursed me for the cost of this. And if you look at it, I'll try to do it. So this is open. Come on. And here's closed. So I can really show the biology of it right here on my arm. A parlor trick, if you will. Thank you. Okay, where were we? Tattoo. Okay, so, we found the thing. What does it do? So we've used both animal models to see what they do, and indeed, without this protein, animals do not sense touch. They do not have proprioception, means they're uncoordinated. And then our colleagues, Alex Chesler and others at the NIH, actually identified individuals who have mutations in the piezo protein. And very interestingly, these individuals, just like what we've shown in the mice, they have severe deficit in touch sensation and proprioception. So this is one such individual. She's blindfolded, just like Yuki, but she doesn't have piezo2, and she's simply asked to walk in a straight line. As you can see, she has major issues doing this because of this lack of proprioception. Happy to talk about it later. Clinically, this is so interesting because doctors are not used to sensory deficits. And when this individual, for example, went to see doctors, they just told her that she must have muscle problems, motor neuron problems. It's not just you. Even doctors don't know about proprioception. And so, but there's now about 100 individuals who've been identified worldwide that have this deficit. And so it's becoming a new syndrome, and we're learning so much more about the importance of proprioception and hopefully how we can treat it better. So piezos, over the last 10, 14 years we're studying, they're doing so many things. But everything has to do with pressure sensing. All those things in your body that happens because of pressure or tension seems to be mainly through these ion channels. So I talked about touch and proprioception. We've shown that some form of pain is through piezo2, and this is called tactile allodynia. Two complex words. All it means is that when touch becomes painful. So you experience this, for example, after sunburn or injury, but this is something that folks suffering from neuropathic pain experience all the time. So very important clinical manifestation. Mechanical itch. If you have bug crawling on your skin or wear a sweater that's very itchy, that's through piezo. So every time you experience these things, please think of piezo. Another thing I quickly mentioned in the beginning is this idea of interoception, which is sensing of internal organ, your internal self. And these are also piezo-dependent. We found, for example, that every breath you take, you're sensing how much your lung stretches, and with this information, you're controlling your breathing rate. Baroreception is control of blood pressure. Now, you're completely not conscious of this, but with every heartbeat, your aortas have sensors that are measuring your blood pressure and changing it to make sure you have constant blood pressure all the time. You're not even aware that your body's doing this. Now, this one you're very conscious of. Again, every time you have to go to the restroom, think of me, think of piezo. As your bladder stretches, it's telling you it's time to go. And individuals who have mutation in piezo2 that I mentioned actually have this problem. They don't sense the bladder filling at all, so they go to the restroom 3-4 times a day to avoid accidents. Also for gut motility, and there's lots of new research that we haven't even started yet. I love this drawing by Christoph Neumann, and it's telling you that he's very full. Now, if you eat a lot, you feel full. It sounds very mechanical, but most of research on this is based on nutrient sensing. Nobody really has studied how much this stomach stretch contributes to how much you eat, when you eat, diabetes, obesity, etc. And me and our colleagues are now studying this to understand and have a new way of understanding and treating diseases associated with this. So everything I told you till now is about humans and animals, but remarkably, piezos are actually present in almost every species we know. They're present in even unicellular organisms. They're present in plants. And one of the studies that we did, actually, we found that piezos are expressed at the tips of plants' roots. And then they are actually required there to sense how rough the soil is and to navigate the soil through that. So just imagine evolution, once it has found this sensor of pressure, it uses it for you to sense touch and for plants to sense the dirt that they're, the soil that they have to penetrate. I find that quite an amazing example of evolution. So that's all the science I was going to tell you today. Also, I feel it's a bit presumptuous that I'm giving you some lessons I've learned across the 25 years of research that I've done. And some of these, I think, mainly apply to scientists, but I actually think that anyone can potentially benefit from this. And all of you at Davos, are people who have lots to do all the time. And this is my favorite concept that I adhere to very strongly, and that is don't be too busy. I feel like I'm not creative when I'm busy, and so I create the time for myself to be able to think, read, and not just do busy work. I have meeting-free Tuesdays, for example, no meetings on Tuesday. I love it. Change fields. I've changed fields a lot during my career, and I think it's to do something else is so important because it brings a new experience. You bring your experience, new perspective to new problems. And you can look at this concept in as wide of an angle as you want. I'm also an immigrant, Armenian, born in Lebanon, came to U.S., and I think immigrants bring so much new perspective with them, and so big fan of that. It's kind of funny in a list of advice to tell you don't listen to advice. But I also find this very useful because, in the beginning of my career, you get so much advice. And sometimes when you hear it from distinguished people like all of you here at Davos, you feel like you have to listen to them, but you don't. It has to make sense to you. Surround yourself with critics. This is so important, especially if you're in a place where people are always praising you and saying nice things about you. It's very important, and I don't seem to have a problem with this because all my family and friends are very eager to criticize me. And I think the last one is, for me, I could say that I got into science to cure diseases, but the reality is I did it because I love it. It's fun. And I think for many of you, probably you do what you do because you think it's fun. But I also realize that sometimes, the little details take over and, oh, my grant was not funded, my paper is not accepted. It kind of gets to you and you stop, you pay more attention to those or not. So I like to remind myself once in a while that I do this because it's super fun and just kind of ignore the annoying details. Anyway, thank you very much for coming to this session, and I think now we have time for some Q&A with Irene.
Great. Well, hello everybody and welcome to this session. We definitely will have time for some of your questions, but before that, let me just first of all on your behalf, and once again from me as a fellow pain scientist, congratulate Ardem, not just on winning the Nobel Prize, deservedly so, but just on a fantastic presentation there. This is a really tough subject, and your skill at presenting it in such an accessible way is absolutely awesome. So thank you so much for that, and also to you, Keith, for that stellar performance. Absolutely wonderful, so thank you.
Thank you. Thanks for saying that.
I didn't know you had a tattoo, so the burning question, literally, I have is how painful was it to get that done? Because that's a huge, huge tattoo. I'm just wondering.
It took 6 hours. It was painful. And, different parts of your body are more painful than others. And this, where the cap of the protein is quite painful. But, as I was getting it, I was totally amused by the fact that what I'm sensing is partly because of what's being tattooed. So there are a lot of interesting parallels.
A lot of symbolism in having it done. It's a very cool thing, and it works really well. I think as a demo, it's a fantastic thing. Ardem, considering how important proprioception is, as you very clearly detailed, it strikes me as odd that it's taken this long for us to discover it. Why do you think it has been such a challenge? I mean, it goes back to sort of that need to persist in science and having the fun with it and keeping going. But it is, striking that it's this is a recent discovery.
Yeah.
And so what were some of the hurdles? And why did you particularly want to go into that space?
I mean, I think about this a lot. I think the other senses, vision, taste, and smell. I don't want to get into too much technical detail, but because they're in one place, there's a lot of material for neuroscientists to take and tease apart. They also were in a group of proteins, GPCRs, that are very homologous. So you can, if you find one, you find ones that look like this. Mm-hmm. This, as I said, you saw those neurons extending. They're all over the place. You don't have easy access to it. And we didn't know what this was going to look like. Yeah. So in the genome, we have 20,000 genes. And we had no idea which one it is. 1 in 10 are membrane proteins, but that's 200. So it's very, very difficult, 2,000. So it's very difficult to kind of parse it out. In science, a lot of times, discoveries happen when the unknown question starts overlapping with technology that's available. And in this case, molecular biology has come a long way, and there was this technology called RNAi where you can knock down one gene at a time and see what happens. And we didn't have this technology before, so you couldn't do this cellular experiment that we did.
Yeah.
But yeah, the general idea is that for any young scientist listening to this and picking a question, pick the biggest unanswered question, but with the practical idea that you should have the tools to address this in the next 5-10 years.
Yeah, it's a really key point, particularly those maybe listening online as well. You've presented the one structure, and you've obviously got it, forever emblazoned on your arm. That's a vulnerability, though, in biology. So presumably there must be some what we call redundancy or variations of a theme. I'm just interested for the audience and listeners online to learn how many different types of that are available, so that if you sort of had a genetic mutation and one was knocked out, you could see from that video just how devastating that would be for humans. Really worried about what would happen in plants, considering food security issues. So could you bring to life a little bit the work you're doing to understand maybe the question about how resilient are we with maybe a few variations of those receptors?
So we have 2 piezos, and it seems like piezo 2 is mainly in neurons, but we're finding that piezo 1, the sister molecule, is expressed in many different cells, in immune cells. And one of the very exciting direction we're going is, the way I introduced it, we knew there was all this pressure sensing in the body, and then kind of what I call the first stage of our discovery was to say, yes, piezo 2 is responsible for touch, for this type of pain, etc. Now we're finding completely new areas of biology that are dependent on pressure sensing. Immune cells, macrophages, eating up red blood cells, for example. Nobody really thought about that being a mechanosensitive, very important immune function, but we showed that that's indeed the case. This and many other cases, I think in 10 years, we're going to look back and say, we had underestimated how important physical sensing is to our physiology and usually diseases.
Yeah, no, I can see another Nobel Prize coming, because there's a lot more discoveries yet to be had. And it's sort of on a systems level, which is sort of where I work at the sort of brain end of things, that importance of touch, as you say, the communications between the immune cells, different roles for it that we wouldn't understand. It sort of emphasizes the community sense, and why do people turn up in freezing cold Davos and slip around on the ice? It's because we want to be together. So whether at a molecular level all the way through to a sort of a community level, I think there's some really interesting points there about the importance for biology and life to have touch and to communicate.
I totally agree, and I find the fascinating is how you can start with the molecule. It kind of opens the door to understand bigger questions of biology and physiology.
Yeah,
and
no,
we're having a great fun doing it.
Yeah, and just slightly left field question: what happens in space when there's no gravity? What's the sort of cost? Because often they say when they come back, if they've been out on the space station, they have some trouble walking. They talk about proprioception being gone. Are they right?
It's actually, this is not our work, but this is actually also piezo dependent. So astronauts who go into outer space, and if they stay there without gravity for a while, they come back with reduced bone density, which is of course not good. And in mice at least, it's shown that if you don't have piezo 1 in the bones, you have the same thing without having to go into outer space. That bone density is controlled by. So the constant tension in the bones is really required for this. And this bone density and pressure works both ways. For example, professional tennis players who use one arm much more than the other have higher bone density in their dominant arm compared to the other. Yeah. So it's just, again, another example of, you don't think about it all the time, but pressure and biology, there's lots of links there.
Yeah, really interesting. And before I open up for questions from the audience for the last couple of minutes, what are the therapies that can come for those sort of rare conditions that are lacking the piezo? Are you working in that space about sort of genetic engineering?
Absolutely. So one of the trouble with, it's so hard to make drugs for various diseases. And as I said, there's a need for pain medication. And there's been many, for example, that they are developed in animal models and then don't translate to humans. What's good about this, what we call target, is that I showed you we have human validation, proof of concept that it will work. The concern here is that although tactile allodynia, we know will be, in a way, ameliorated by blocking Piezo2. Obviously, it's not a drug that you would want to take orally and get rid of all your touch and proprioception and pain. Nobody wants to do that. And so we're trying to develop small molecules that would block Piezo2, but it has to be in indications where it could be used topically, like only on your knee, on your elbow, for example, so it doesn't interfere with your normal body functions. Yeah. It's early stage. It really takes another channel, TRPA1, that my lab cloned out in, there's phase 2 clinical trials on it. We found it 20 plus years ago. So it's just, there's so much work to be done from the basic biology to translation.
Yeah, that's just a long journey. Well, it's time for you to have the opportunity to ask Ardem any questions. So we have a roving mic. If you could just put your hand up. Please, the gentleman at the back.
Thank you. That was amazing in so many levels. I'm a physician, and I was wondering, outside of patients who have an obvious neuropathy from nerve damage, there's patients who have actually hyperpathic syndrome and pain and all that. Is that an hyperexcitability of those receptors, or it's a decrease in a threshold? What is the mechanism?
There are other genes, including TRPA1 that we've talked about, where its hyperexcitability causes pain syndromes. We haven't found any that's associated with piezos yet, but that doesn't mean it doesn't exist. There's very exciting databases now of genetic data associated with your health record. UK Biobank, US is starting one called All of Us, and these kind of databases will really help us connect what conditions are associated with what genes. So work in progress.
Fantastic. I'm just going to ask the organisers, can I take another 2 questions? Is that okay? I know online have gone off, but it'd be great to have one more question. One more question. I think was there somebody on this side? Hands up again, those who'd like to. Okay, gentleman in the front.
So. Hi, and that was great, thank you. Do different people feel pain differently? So if I put my finger on in hot boiling water, and you put your finger in boiling water, is it different?
Absolutely. I think there's what we call neuroscientists as internal state, which really dictates. So you're not automatic, electrical wires. I gave the thing, but it's not quite like that. I'll give you the easiest explanation. You're outside in Davos, you're super cold. You go take a warm shower, it feels so nice. But if you went on running on a hot day and you come and take a shower at the same temperature, you'd find it painfully hot. And so within your individual, the same stimulus is different all the time. So there's no question that we all experience things slightly differently.
Yep, context. Oh, I'm so sorry we've run out of time. But Ardem, I'm sure you're willing to stay for a few moments, either here or outside the room, if there's somebody else coming in to take a few more questions, because I can sense the hands up. Thank you all for coming. Ardem, thank you so much for giving a brilliant presentation. Congratulations once again.
Thank you.
And to Yuki as well.